include prednisone and Acthar gel, which are indicated for under connective tissue disease umbrella, and myositis is part of that. So, but you cannot run FDA-approved treatment in myositis. So first of all, I wanted to show you a little bit of the progress that we have made in the field of myositis, because I'm showing you two graphs here. One is before 2009 and one is since in last 10 years, what have we done? And if you can appreciate, we have doubled the number of clinical trials in last 10 years in polymyositis, dermatomyositis. So that's a progress to me. But the question is this progress enough? And I often ask, is this enough? Is this going to get us the answers that we need? So to answer that question, I often look at a disease where we have made significant progress and really change the way we actually look at the patients currently, and that disease is rheumatoid arthritis. As you know, rheumatoid arthritis used to be a very bad, I mean it still is, but used to be very, very bad, severe, disabling, painful, deforming disease, where patient will go from early rheumatoid arthritis to late deformity, severe deformities and pain. And I can tell you, when I started my training in 2003, 2004, I did see some of these patients. Today, it's a rarity to see any of the patients with that kind of joint. So in 15 to 20 years, we have made a huge progress. The reason I go to rheumatoid arthritis is I say, okay, what can I learn from there that I can apply in myositis world so that we can make similar progress in next 5 to 10 years? And you know how many FDA-approved treatments there are for rheumatoid arthritis? There's a whole laundry list of the treatment that are approved for rheumatoid arthritis. In fact, it's so difficult that when I have a patient with rheumatoid arthritis, I can't even decide what drug to give. I can almost, flip a coin, or I can't even flip a coin. I have to, play with the cards to pick what drug should I give for my patient next. So that's the kind of success we are talking about. And to be honest, we see patients here, or they are normal. We don't see this, or we don't see this anymore. And I can tell you, when I see my patients with rheumatoid arthritis, oftentimes I'm confused. Did I really make a right diagnosis? Because I can't even detect any rheumatoid arthritis few years later. So, and when I get patient referred from other doctors because they leave or patient move, their insurance changes, I said, I'm not sure you have rheumatoid arthritis because you don't have any evidence of rheumatoid arthritis. Let me go back to your notes. And sure enough, they had rheumatoid arthritis. It's just I can't detect it anymore. So that's the kind of success we're talking about from a disease, for a disease that used to be thought to be almost like disabling disease and no treatment available. So how did they reach there? They reached there because they did many clinical trials. You're talking about 455 clinical trials before 2009, and in the last 10 years, they've done 600 clinical trials. So it takes a lot of doing to find the right drug that will work for a disease. Now, a lot of people tell me that rheumatoid arthritis is a wrong example to compare because myositis is a rare disease. So how can you do hundreds of clinical trials in myositis for a rare disease? There are not that many patients, or it's not going to be possible. So I tell them, look at vasculitis. Vasculitis is as rare or even rarer than myositis, and they have done 100 clinical trials in last 10 years. That's about 10 trials in 1 year. So that's pretty impressive. And what did they get in return? They have 2 FDA-approved treatment that works in this disease. And so to me, that the argument that rare disease can't achieve that is not a good argument because I have a disease that I treat that actually has achieved it clearly. Now, it's not, nothing starts with clinical trial. It all goes back to this slide. I don't want you to understand this slide, but the whole idea of this slide is this rheumatoid arthritis pathogenic pathway slide. So essentially, these are all key players in immune system that are playing different roles in inflammation of rheumatoid arthritis. And over the years, researchers, basic scientists understood these pathways, understand what's connecting to what's stimulating what's inhibiting what, and that's the whole process that led to saying, aha, if we block this, or this guy here, or this guy here, maybe we can achieve some improvement. And that's the process that went in the research 10 to 20 years later that we have effective treatment. So it takes a lot of doing. And we are fortunate enough in myositis now that we have really good, dedicated basic scientists, clinical scientists who are doing this kind of work, who are trying to make pathogenic pathways of what affects what and what lead to, then ultimately leading to the muscle damage, what's affecting the muscle, what's the downstream effect of all the immune cells here that is affecting muscles. And then we are also starting to block some of these important molecules with different drugs and trying to see the results. One of the biggest studies we did is rituximab, where we block this B cell completely and see what are the effects on the muscle or myositis in those patients. So we have started to do that, and we are making some progress. However, myositis is very difficult because if you think about myositis, it's not single disease, it's multiple different disease. Somebody has polymyositis, somebody has dermatomyositis, somebody has high inclusion body myositis. So various different subsets of disease makes it difficult to study. Also, myositis is not a single organ disease. So not only it has various different subsets, also each subset affects different organs for different patients. So it can affect lungs, for some patients it can affect swallowing, it can affect skin, it can cause arthritis, Raynaud's, and so on. That again makes it very difficult and challenging to study and do clinical trials. And also we have recognized that myositis itself has various different autoantibodies. So these are sort of the blood markers that any patient would have. And nowadays we can tell based on the blood marker of a myositis patient that what clinical course they're going to take, what kind of problems they're going to have. So now should we study a particular clinical subset defined by this autoantibody? And we have started to do that. In one of the trial I'm doing, we are only studying this big subset called anti-synthetase syndrome. So we started to do that, but it gets it complicated. It's a rare disease to begin with, and if you use autoantibodies, which are very helpful, but then you're making it rarer. So again, it adds again to the complexity of the clinical trials in myositis. And also we have various different pathology. If you look at the muscle biopsies of polymyositis versus dermatomyositis versus necrotizing or IBM, they look like completely different diseases. So how can you study them together? So it makes it again challenging. But all of these challenges are fine, but again I go back to a disease where there have been success, and what's more complicated and heterogeneous and has more subsets than myositis is vasculitis. So to me, they have made this, they have been successful with 100 trials, 2 drugs approved. We can achieve that. It's much more complicated. Vasculitis is far more complicated than myositis, affect far more different organs, have far more subsets. All the things that I've told you has far more different pathologies. All of that is still possible and achievable. So let me explain you what the progress we have made in last 20 years, and especially in last 10 years. So first of all, to do any clinical trial, you need to have a uniform diagnosis, uniform way of diagnosing, where everybody, rheumatologist, dermatologist, neurologist, pulmonologist, agree that that's the disease that we're going to call dermatomyositis. That's the disease we're going to call polymyositis, and so on. So that process is called classification criteria, a criteria of the disease. And there were previous criteria, but previous criteria had problems. And over the last 7 years, since 2010, this project started, which is called is classification criteria of adult and juvenile idiopathic inflammatory myopathies. And I was very well involved with this project, and it took 7 years to finish this project. So it takes a long time to do these things, but it was a very intensive project. So there were about 40 to 50 centers around the world, and many of you contributed as cases and controls for this study. And then there were about 75 clinical features studied in this study, and then led to another validation in 2 different independent cohorts. At the end of the day, we now have a criteria that almost all clinical trials in myositis can use to define what's polymyositis and dermatomyositis. But again, it takes a lot of research, a lot of patient contribution, a lot of patient participation, their samples and tissue, and then we finally come to a criteria. So this is a criteria. I don't want to even go over the criteria, but the idea is we give different scores to different clinical features of the patient, and if you have enough clinical features, then that's what we call as a disease, a particular this or that disease. So the other problems that we had in myositis clinical trial realm is we didn't really had great outcome measures. And the work that started by Lisa Ryder and Fred Miller in 2001, where they said, let's come up with a consensus amongst all the myositis researchers, what should we study in any myositis patient in a clinical trial or a study. So what should be the measures? So outcome measures are something that is basically used to define a patient has improved in a trial or a study or not improved. That's called outcome measure, because to do a scientific study, you need to come up with a criteria who we will call improved and who will not call improved, and that's the process. So it started in 2001, and they came up with 6 core set measures. The core set measures are 6 measures that we're going to do on the patients to understand how their myositis is doing. And that we will use then to define is the patient improved or not improved. In 2004, they come up with the consensus of improvement. And what it was these 6 core set measures, and I would like to explain you in a bit. Basically, it's called Physician Global, which is a scale of 0 to 10 centimeter, where a physician basically marks how severe they think somebody's myositis is doing. So that's one criteria. Then there's a Patient Global, where patient marks on a scale of 0 to 10 how severe they think is their myositis, how active is their myositis, what is their perspective on their myositis. MMT is manual muscle testing. Most of you know that your doctor evaluate your muscle strength, and that was the MMT, manual muscle strength testing. So how strong our overall muscle groups are. Health assessment questionnaire is actually patient-reported outcome, where you are asked about 20 different questions, where things like, are you able to brush yourself? Are you able to get up from chair easily? Or if it's not, then how difficult is it for you to get up from chair? And so it's kind of measures your day-to-day activity and comes up with a score or a measure of outcome. Then muscle enzyme, many of you may have high muscle enzyme when you were diagnosed, and that's another criteria that we use in clinical trial. And last but not the least is called extramuscular disease activity, where physician rates not the muscle disease, but everything else than the muscle, like skin, lung, joints, everything else that affects you, fatigue, those kind of things. So these are the 6 criteria that we now use in any myositis clinical trial to define a patient improved or not. And earlier criteria there was that you have to show improvement in 3 of these 6 measures to call somebody improved. However, those had some limitations, and the limitation was where you could only say somebody is improved or not, you can't say somebody is majorly improved, moderately improved, minimally improved, and so on. So in 2016, we came up with this criteria. Even this project took 5 years. We came up with a better way of scoring those 6 measures, essentially, where we can have actually a good outcome of saying who's minimal improved, who's moderate improved, and major improved. And that way it can help in clinical trial to decide whether this drug worked or not worked. So I think also we need to still do better outcome measures for our trial. We have some outcome measures, we have a classification criteria, so stage is set, but I think we need to do a better job. And I'm investing a lot of my time in understanding how can we do better job in measuring our patient's outcome. For example, Fitbit, physical activity monitors, is one way to measure patients' outcome, where patients could, when they're weak, they're not walking and doing things that much, but when they get better on treatment, they might be walking better. So could we use that as a measure of outcome? Patient-reported outcome, I've explained you what can we design better patient-reported outcomes that are more meaningful to the patients. Dynamometry is an instrument that I use to measure muscle strength, which gives me an objective number of muscle strength rather than me just subjectively saying is he or she strong or not strong. Functional tests, we ask patient to get up from chair, walk about 10 feet and come back, and how long does it take for them to do that can be a measure, actually a functional measure of how patient is doing. So on and so forth. So we are investing a lot of time in improving our outcome measures that can improve the quality of the clinical trials that are going on. I want to also explain you the drug discovery or drug approval journey. Basically, what happens is it all starts with the molecule. So first you need to find the target based on the pathogenic pathways I've shown you. So if you find a target, then it leads to animal testing, which is the mouse model we keep talking about, different mouse models and different studies. And then it led to an application to FDA that we want to do this, study this drug because it worked really well in a myositis mouse model, so we want to check it on the patients. And then that first step of it called phase 1 trial, where basically it's healthy volunteers who would volunteer to be checked for that particular drug to make sure there's no major harmful effect, because we don't want to test the drug directly on the patients. We test it on healthy volunteers. And then if there's no safety issues there, then it goes to what we call as phase 2 trial, which is a smaller trial, proof of concept type of trial, where it's you're looking for safety, you're looking for some signal of improvement. So it's not a definitive trial, but enough for us to get to the stage 3, which is what we call as phase 3 trial, which is what we say it's a definitive trial. So if you have a phase 3, which is positive, then you can go to FDA saying, hey, this drug phase 3 trial was positive, so that means we should get an approval for this drug. And then there's also a long, even after that, there's a long process of the finalization and getting to the market. And then after that, there's called phase 4, which is sort of a post-marketing. After the drug is available to the patient, then there's also more studies going on where to understand what are the risks that we didn't discover during the trial. Maybe when we give it to hundreds and thousands of patients, we'll discover there's some problems in the drug. So that's this whole process takes about 10 to 12 years and takes anywhere between $100 million to about $1 billion. So think about, and if there are about 10 to 100, it takes about 10,000 to 15,000 compounds, and only 5 are finally tested in humans. So here, from the lab to human testing, only 5 comes out, and amongst those, only 1 get approved. So from 10,000 to 1. So that's why drug discovery process is rather complicated. Companies spend billions of dollars into doing this. Many years goes by for academic researchers and drug companies to ultimately come up with a drug. So it's a very difficult process. I hope you appreciate it. It takes a long time and effort. However, I'm very happy to report that we have made some progress in myositis in the last few years. This is just a graph to show you phase 2 and phase 3 clinical trials in polymyositis and dermatomyositis over the years. So from 1990 onwards. So if you look at last couple of years, we used to do only 1 trial a year, and that's a very ineffective way. And in last few years, we are starting to do 2, 3, 4, 5 clinical trials starting in a year, which is very good for myositis patients, because more clinical trials you're going to do, more are the chances for you to find the drug that works for the patient. And honestly, even if we have one drug which is approved and works for patients, that particular drug is not going to work for everybody, because I've already showed you how different the diseases are within myositis. So we need multiple drugs to get approved for myositis to cover large range of patients. So the good news is that we are starting to do multiple clinical trials. Here's the myositis pipeline. I'm again happy to report that pipeline looks better and better, looks rather robust. We have multiple clinical trials that are going in phase 3, phase 2 trials. So we are very happy that many drug companies are coming up and trying to look at their product in dermatomyositis and polymyositis. The other thing I wanted to tell you is clinicaltrial.gov is an excellent resource for every patient, everybody. It's not very patient friendly, but still it gives you the right information. So if you go to clinicaltrial.gov and you put in your disease category and you put in the country and hit search, it will show you the list of all the clinical trials that are ongoing in your particular disease area. And if you click that particular trial, then generally at the bottom there's a list of centers that are participating in that clinical trial. And if you're interested in participating in clinical trials, then you can make a phone call to the coordinating center or a coordinating coordinator to see if they would be able to take you in clinical trial. Again, as I always appeal patients to join the clinical trials and clinical research, because as I've hopefully shown you, how much does it take to get from A to the drug that is approved. So it takes a lot of effort. Even in classification criteria, response criteria we did, it took thousands of myositis patients to get there over 5 years. So again, if you don't participate, we won't get there. So I think that's the pitch I wanted to make. But anyway, so now I wanted to show you some of the promise that we have in next few years. So these are the 4 drugs that are currently in phase 3 clinical trials. One is linabecestat, IVIG, sub-Q IG, and abatacept. So let's first talk about linabecestat. So this is a drug which I like to call it as anti-fibrotic, anti-inflammatory drug. It's a little difficult for me to explain you its exact mechanism of action, but it blocks certain receptors that are on immune cells that leads to anti-inflammatory properties of this drug. And this drug is basically has had 1 trial so far on dermatomyositis patient, and I like to explain you this graph. This graph is C-DASI. C-DASI is just a severity of skin disease. So that's on this axis, and this axis is time. So the orange graph is where patient got placebo, means they did not get drug, and the blue ones are the where patient got drug. So if you can appreciate that the blue and orange separates out very well over the 16-week time period, that basically lower score means less severe skin disease. So they all started with severe skin disease, but there's a significant decrease in the skin severity in linabecestat as compared to placebo. Then they did an extension phase of it. So they then continued these patients, and if you see over the longer period of time, their skin continued to improve. So to me, this is a very positive pilot trial or phase 2 trial. And that trial is led to a phase 3 clinical trial now on the drug, where there's a randomized control trial for 52 weeks with 2:1:2 randomization, which basically means if there are 5 patients, 2 patients will get 20 milligram dose, 1 patient will get 5 milligram dose, and 2 patients will get a placebo. It's being done by Corbus. They have about 75 sites or 30 sites in US, and they're only evaluating dermatomyositis patient. However, adult juvenile dermatomyositis could be considered as well. Their outcome measure is the new outcome measure that I already reported to you, and their goal is to enroll about 150 patients, and the trial is currently active, and as my understanding is there are already patients enrolled in this trial. I don't know what the number is, but it's currently active. Next, I'm going to go to IVIG and sub-Q IG, SCIG. So IVIG more than sub-Q IG has been used in dermatomyositis more than polymyositis for several years. Since I remember from my residency and fellowship years to about 15-20 years ago, it has been used since then. And we think that this drug works, at least in certain subset of patients, this drug really works. The problem is we can't get this drug for all the patients because it's not FDA approved. So I'm very happy that there are companies that are coming forward to do clinical trial on this drug to actually get an FDA label on this drug if it gets approved. And there are various different ways IVIG or immunoglobulin can work on our immune cells. So we actually don't understand exactly how it works on immune cells, but we know that it does work on autoimmune diseases and definitely works on dermatomyositis. So as an evidence, this study was done in 1993 by Delakas et al., and there were about 15 patients in this study. So what they did is half the patient they gave drug early. So they gave about IVIG to half the patient. The patients and this is an MRC score, which is a strength score. So half the patient who got drug early got better in 3 months. Then they gave placebo to those patients. The patients who got placebos, many of them worsened in the next 3 months. Half the patient gave placebo first. When they gave placebo, they saw either worsening or not significant improvement. But then when they gave IVIG to the same patient, they significantly improved. That's a very good evidence that the drug may work, but it's a small study. Again, it's a phase 2 type study that gives you an indication, but it's not definitive enough or not rigorous enough for FDA to give you approval for this study. However, that study was then followed by another study where there was a mix of polymyositis and dermatomyositis study, and unfortunately that study did not show a difference in manual muscle testing, which is muscle strength, or muscle enzyme, which is CK. The critique of that study was it did improve various other measures that was being done on the patients. So was it a good study? Was it a failed study? Did the drug fail or the study failed? There was sort of up in debate. So that has led to this large clinical trial called ProDRUM study, where it's IVIG, where they are giving 2 grams per kilogram IVIG every month for 4 months, 16-week trial, and then after that everybody gets drug for 24 weeks. So for 16 week, half the patient get drug, half don't, but then after 16 week, everybody gets the drug. And this is being run by Octapharma, 49 sites, and it's only for dermatomyositis patient. And I'm happy to report that this trial has been finished. They have completed the enrollment. Their last patient should be in the last patient visit should be ending soon, and we might see some results end of this year, early next year. So I'm really hopeful for this trial. So some of the advantages of IVIG is that it's not immune suppressive drug. So it can be used for infections, pregnancy, patients with cancer. So that's a huge advantage to use IVIG. And also, it's strongly efficacious in dermatomyositis, specifically in patients who have skin rashes, patients who have difficulty in swallowing, and also seen to be have improvement in juvenile dermatomyositis patient. IVIG or sub-Q IG has some limitations or problems. And one of the biggest problems in early on when this drug was being used is blood-borne diseases like Hep B, Hep C, HIV. The good news is that's not a problem anymore because they have a process in which they can actually eliminate the bacteria before the drug even gets to the market. So that problem is not there anymore. However, the issue with IVIG and sub-Q IG, there's still some reactions when you give the drug, and 5 to 15% patients may get reaction. And the most adverse events that I see with IVIG is headaches. Generally, that can be very disabling for a patient and they would have to stop the drug. And also, the other problem with IVIG is very cumbersome. It takes a lot of time. You have to go to a center to get IV infusion for 1, 2, 3, or 4 days every month. It's very 4 days out of a month gone. It's very difficult for many of our patients who are working. So some of the less common side effect but serious side effect could be aseptic meningitis, congestive heart failure, renal failure, blood clot, and so on. The reason I'm trying to tell you more about the side effect is because I believe so many of these side effects are due to a lot of volume that goes in a patient at once. So if I give this drug in 1 day, it's a lot of fluid and proteins going in patient's body all of a sudden. And what happens is that lead to many of these side effects. Whereas if I have to just eliminate the side effect, I can slow the rate and give the same drug over 5 to 10 days and there will be maybe no side effect then. So because the side effect is dose dependent, one of the thought process has been that what if we give the same drug as sub-Q injection and have the patient take it at home, and so prevent this logistical problem for patient to travel all the distance and get drug for 4 days in a month. And there are companies, they're actually now sub-Q IG available in the country. It's not approved for this disease, but has been approved for some other diseases. And we are fortunate enough that we are actually, this is just a picture for how the sub-Q IG works. There's a pump that you get and you have to self put it, it's a sub-Q injection, so you have to put a needle under your skin and the drug gets slowly infused over the weeks. So, it's only a couple of hours a day and a couple of days in a week that you have to infuse this drug. And the reason I'm telling you all about sub-Q IG is now we have a clinical trial that is starting with using sub-Q IG in dermatomyositis. And this is a randomized controlled trial for over 24 weeks. So that means half the patient will get drug, half won't for 24 weeks. But following 24 weeks, for 28 weeks, everybody will get drug. This is being run by CSL Behring, and there are about 94 sites, and the dose is about 0.5 gram per kilogram sub-Q injections every week. The study is being done in dermatomyositis, adult juvenile dermatomyositis, but also in amyopathic dermatomyositis, means the patient who don't have muscle weakness but have significant skin issues. Even on those patients, this study is being done. Again, the same criteria is being used, and there are about 126 patients to be enrolled in this study, and I'm happy to report this study is active or going to be active rather very soon. Another drug is currently in phase 4 is abatacept. This drug basically is an approved drug for rheumatoid arthritis for last 10 to 15 years now, and has been seen very effective in rheumatoid arthritis and other arthritic conditions. And the idea was that it actually blocks the interaction of two immune cells, key immune cells in our body called B and T cell. And because of that, it cuts down the inflammation. So since myositis patients get inflammation, so idea was that could we use this drug to cut down the inflammation of myositis patients. And there's a study done by Ingrid Lundberg out of Sweden, where she actually showed very nicely in a study that this drug may work. Again, this is kind of phase 2 study, cannot be proven, but it gives an indication this drug may work. In this study, what they did is they gave active treatment to half the patient early, and then half the patient did not get anything for 6 months, then got active treatment. And their idea was the patient who got drug early would improve earlier. And that's what they showed in this graph. Basically, the patients who got drug early, this is the black bar, they improved significantly within 3 months. And after that, they kind of plateaued. But the patients who didn't get the drug for 6 months did not improve much. But when they did get drug at 3 months, they improved again significantly. So again, that showed good evidence that this drug may actually work in polymyositis, dermatomyositis patients. And so this trial, this is a phase 3 trial, which is an ongoing trial, is again randomized controlled trial for 24 weeks, followed by open label extension for 24 weeks, which basically again mean half the patient get drug and half don't for 24 weeks. Then for next 24 weeks, everybody gets the drug. The good news here is that many different forms of myositis are included: polymyositis, dermatomyositis, necrotizing myopathy, overlap myopathy, even juvenile dermatomyositis is being studied in this trial. And all adults, the target is 150 patients to be enrolled, and I think they have reached about 50, 60 to 70. They've reached about half the target of where they need to be. So we probably should be seeing some results next year on this trial. So now going from phase 3 to phase 2, now these are 4 trials. We hopefully will get the results of these trials in next several years. Some are sooner, some later. But what are some of the phase 2 trials? Because phase 2 trials are the trials where they can hold promise that they can go to the phase 3 if they are proven positive. So one of the drugs that I want to talk about is this Kezar drug called KZR-616 drug. Basically what this drug does is we have 2 types of proteins, immune proteins, special proteins in our body. One is which is called proteasome, which are of 2 types, constitutive and immunoproteasomes. The constitutive proteasomes are the proteins that helps to do regular day-to-day functions of our body. So if you can't block those, if you block those, that lead to cell death and problems. So the idea was, what about if we selectively block those kind of protein that maintains the balance of only the immune cells? Maybe the problem is there. If we block that, could we see some results? And that's the idea that led to this drug. Basically, this is a drug. This is a melanoma cell. Melanoma is a type of cancer. A myeloma cell. Myeloma is a type of cancer in which you block both the proteasome, the regular one and the special one, which is only on immune cells. If you block both of them, it leads to cell death, which is good for cancer patients because that's what you want. You want the cancer cell to die. So if you block both, that can lead to cancer cell death and can lead to improvement in cancer. But we cannot do that in our immune diseases. So if we can selectively block only these special proteins that are on immune cell, then it may lead to decrease in the inflammatory, what we call cytokines and chemokines, which are the signals of inflammation, and that may lead to decrease in inflammation and hopefully improvement in some of our patients. So that study is being done. It's not active yet, but it will be active soon. It's a randomized controlled trial for 16 weeks, followed by a crossover design over next 16 weeks. Basically, half the patient got drug and half don't, and after 16 weeks, they switch the groups. The patient who got drugs, treatment protocol A go to B and B go to A. And they are using, and the good news here is they are studying both polymyositis and dermatomyositis, again only adults, no juvenile, and this trial is not currently active but hopefully will be active soon. Tocilizumab, which is anti-IL-6. IL-6 is again very pro-inflammatory molecule in our body. So idea was, and it's an approved drug for rheumatoid arthritis. So the idea was if we block this very pro-inflammatory molecule, that may downstream decrease the inflammation. And that's the idea of this drug was, and we are happy to report that this is a trial being done from our center, and we have completed this trial and actually currently analyzing the results. I don't know the result myself, but we are currently analyzing the results, and hopefully, I'm hoping by end of this year, we'll know the results of this trial. And this was a 40 patient, a small phase 2 trial. We only enrolled about 35 patients, and then it was enough for us to stop the study there. It was 24-week trial over the, again, randomized controlled trial. It was run by Genentech. About 7 sites, including ours, was involved in this. Again, this trial is completed. We should see some results by end of this year or early next year. Another drug I want to talk about is Adrenocorticotropic Hormone Gel, which is commonly called Acthar Gel. This is a very unique situation where we have a drug that works on through various different immune mechanisms. And the history of this is that this drug in 1950s, this drug has been available since 1950s. So this drug was being used before, this is even before steroid was available. So this drug was being used to treat rheumatoid arthritis or gout and other conditions, and it really worked in those conditions. But when prednisone came in picture, then people started using prednisone because it was more effective, and kind of this drug kind of died after that and wasn't being used that much. But a couple of companies picked up on its properties and said, okay, why don't we try this on immune-mediated diseases and try to study it now. And what's the funny part of it is in 1952, this drug got FDA approval for all connective tissue diseases. That includes myositis. So technically, this drug is an FDA-approved drug for polymyositis and dermatomyositis. However, there's no clinical trials that were done on polymyositis and dermatomyositis. This is like really long time ago when the drug approval was very different. So it's a very funny situation where you have a drug approved for polymyositis and dermatomyositis, but not a single patient was treated with this drug that to prove this drug works. So this is I talked to the company, and I think it was in 2009. I said, you know what, I see that you have a label, but I'm not going to use it because you don't have any proof. So that led to a pilot clinical trial that I did on this drug on 10 patients, and we showed that this drug does work in about 6 to 7 out of 10 patients. So 60 to 70% patient, it did work, and just trying to show you the level of improvement. So about 60 to 70% patient improved. This is a manual muscle testing of the 10 patients. Now some did not improve, but you can see some of the patients did improve significantly, and some of their skin rashes improved significantly. And I'm trying to convince the company now, this is a sort of a small, it's even before like a phase 2, it's not even phase 2, it's like a pilot trial, but to go to more like a phase 3 or traditional trial, they're sort of less interested. Why? Because they already have FDA approval. So it's kind of, you do trial to get FDA approval, so it's a little difficult situation. But I think the company is understanding the need of research, that the physicians are not going to use it unless until you really prove it beyond doubt, even though you have FDA approval. So, from that, there's another molecule which is called another thing called interferon. I don't know if you've ever heard of this, but it's one of the signal molecule in our body. And if we look at dermatomyositis patient and lupus patient, and we do their gene analysis, what we find is that the patients, when they're active and have active disease, this a signal is really high even from their genes. Means the genes that produce this kind of protein are activated in patients with dermatomyositis a lot, and lupus a lot more than any other immune diseases. So the idea was, could this be causing the problem from the genetic aspect? So if there's a way to block interferon, could that actually help us to cure this disease? And there's only been one study done in myositis in which actually they were able to show that the patients who had more than 15% improvement in their muscle strength, those patients had much better blockade of their interferon signal as compared to the patient who did not have improvement, implying that if you really block this interferon significantly using the drugs that we have, so this is a drug called cefalumab, which actually can block the interferon alpha, that particular molecule or the protein I'm talking about in our blood, and that might actually lead to improvement. And there's also there's some interest currently in the field to see if we could revisit that and see if we can have better drugs to block this particular interferon and hopefully lead to improvement in our patients. Because, and by the way, just to report, the company, the same company, AstraZeneca, did the study in lupus. And what happened is they did 2 trials with their drug to block interferon in lupus called TULIP-1 and TULIP-2. TULIP-1 was negative, so company was very upset about it because they thought such a high signal in lupus and we blocked the right molecule and we blocked it well, why did it not work? But I'm happy to report about a week ago, TULIP-2 was positive. So now it's a difficult situation for FDA and a company and patients what to do, one positive, one negative. I don't know, I'm not a lupus researcher, I don't want to comment much about it, but because of TULIP-2 being positive, I feel like I can again stimulate the company to think about doing the dermatomyositis trial, because the same high signal is seen in dermatomyositis as well. Anyway, so tofacitinib is a drug which basically is a JAK inhibitor. A JAK is essentially again one of the signal. It helps in signaling from signals outside the cells to really go in the cell, signals to change the how the cell is reacting to the outside molecules. So this drug has been approved in rheumatoid arthritis and psoriatic arthritis, and we were fortunate to see a pilot study result used when this drug was used in dermatomyositis patients. And you can see severe rash here and completely disappeared when this drug was used. Again, it's a single open label, means everybody knew the drug, doctor knew they're giving the drug, patient knew they're getting the drug, so less scientifically rigorous, and but it led to improvement in 10 dermatomyositis patients. So that's again pilot trial, not really phase 2, but could we take this forward and study it more and see if we can get better outcomes in more scientifically rigorous way of doing phase 2 and phase 3 clinical trial. And last but not the least, basically it's another drug which has been approved in psoriatic arthritis and psoriasis called apremilast, in which this is out of our center where we actually gave this drug out of desperation, and we for dermatomyositis patient, we called it psoriasis, got the approval for the drug and gave the drug to our patients, and we saw significant improvement. Again, only few patients study, cannot completely believe it because you have to still do more rigorous science. But the good news is that there is some hope. There are new molecules coming in phase 2. There are 4 phase 3 studies going on. So I really hope that 1, 2, or 3 drugs get approved in next several years for our polymyositis, dermatomyositis, necrotizing myopathy patients. Thank you. And also want to thank TMA for this opportunity to come in front of you guys and give a talk on on the drug because I feel like a lot of time my patients don't understand what really goes in the drug discovery and from the molecule to all the way to the clinical trial, and how important it is for patients to volunteer for research or to participate in clinical trial. And I myself am very fortunate that I have very good patients who actually, I have one patient who have participated in 3 clinical trials, and she's like, okay, I want to, because she goes from one clinical trial, she gets better or not, but then within 3 months she comes back, okay, do you have a clinical trial for me? And so just this is very important for patients to participate, but we are very thankful to our patients when they do. Thank you.
Just a second. If there's no FDA-approved drugs for myositis, how come we can use prednisone and IVIGs and things like that?
Yeah, the reason we are able to use it because FDA doesn't regulate that you cannot use it. It's just that we know these drugs work. For example, we know prednisone works. It just doesn't work for everybody, and it has horrible side effects. Same as IVIG, it works, but we don't know exactly for who it will work, and so there are some patients who it doesn't work for, and also the insurance company don't want to approve it. So we have to fight with the insurance company to get the drugs. Sometimes we are successful, sometimes we are not successful. So I think there's a need to do clinical trials and get an FDA approval. And also, we can only use the drugs that have been approved for some other reason. We cannot use a drug that has been not approved or not on market. So our ability is very limited. So let's say if I want to use, Kezar's drug, I cannot use it. It has to be first approved by some other drug, disease, and available in market for me to even apply for the insurance to see if we could use that drug in this disease. So it's a very complicated process. Even though we have some drugs, they are far away from cure for sure. They're far from getting patients to significant improvement. I generally quote that the rate of remission, if cure is not our goal, remission is our goal, the rate of remission in our disease is only about 10 to 20%. So that means we have to do a lot more work to get there. Yes.
Talked about a prednisone replacement.
Yes. So the Dr. Nagaraju talked about prednisone replacement, and you didn't see it here because that being studied in muscular dystrophy, not in polymyositis or dermatomyositis. So they are looking at obviously if that drug gets proven in that particular disease, then we can pick it up and say we can try in different other diseases like polymyositis, dermatomyositis, but that drug is not currently being tested in this disease.
Any chance of when this may happen?
I think he reported that the trial has finished, and I think they are in the stages of analysis, so I'm hoping by end of this year. It takes, once the trial gets done, it takes about 3 to 4 months to get to know the results. It's a little bit of a process.
Some of these drugs are very expensive, like IVIG. The more people that use it, does that mean the cost is going up because of supply, or the more people that'll be using it, or when it gets, say, FDA approval for DM, the cost might come down, so it's wider distribution. Thank God my wife uses it, and it solves her problem pretty much. And thank God the insurance company picks it up 100% now. But I feel uncomfortable with the people that can't pay for it.
Yeah. So the problem is the cost is very complicated. So cost is a very complicated issue, and it doesn't necessarily mean that if the drug is FDA approved, the cost will go down. I hope it does, but doesn't always necessarily mean that, because cost is a process of how complicated is the process to produce the drug. IVIG, the reason the cost of IVIG is so high, it is very challenging to produce IVIG or sub-Q IG. It's very complicated process. It takes long time to come. In fact, there's a national shortage of immunoglobulins right now in the country, but it's a complicated, because of such a complicated process, the costs are very high. Now, sometimes if your volume goes up, the cost can come down, and we have seen some of that examples in rheumatoid arthritis world. Because and also more drugs approved also decreases the cost. Again, it's basic economics, that more competition will drive the cost down because everybody's trying to, say, use my drug rather than your drug. So I think we have seen some examples where there's a drug called biosimilar. So biosimilar are sort of copies of the original drugs, and since the US government and FDA has allowed those to come in the market, that has led to decrease in the cost of the original drugs as well, because the original drugs are saying if we keep the margin so much, then so their cost comes down. So but the cost is very complicated process. Again, it depends on your insurance, depends on what plan you have, depends on your copay. There are so many things that gets involved in the cost aspect, and I'm obviously not an expert on cost because, and it's definitely not in my hands for sure.
Is the shortage on IVIG, is that caused from the raw material, human raw material that it's made from, or is it the process that's shortening it?
Oh, that's a good question. Why there is a shortage? Do you know? Increased demand, so less supply. Supply is the same. The demand has gone up. So you have to open up new factories and new, manufacturing units to get to the demand. That's
what I'm saying. Is it the manufacturing, or is it the resources that go into the manufacturing?
It's not manufacturing.
It's not. So is there an awareness that we can do for the raw material, the human raw material part of it, to increase that, to help on the shortage?
What I know that FDA is talking to the companies who produce IVIG and sub-QIGs to increase the production. I know that because the FDA has actually come out with a statement on their website that they are aware of the shortage of immunoglobulins, and they are in current discussions on how to help improve the production aspect of it.
And I guess that's, yeah, go ahead.
Do you want me to use? So I work for CSL Behring, and we produce IVIG and SCIG. And what we're doing, we don't have a supply problem, but we've been increasing our plasma centers. So people that donate their plasma, are helping us keep our supply. And we're actually been helping people that are having shortages as well. So, but it is a problem for some companies right now.
Tell me, talk about the difference between cure and remission.
Okay. So cure by definition means that there's no chances of the disease process to come back unless the new process starts. So a new process, so for example, a perfect example is infection. Okay, so if you get an infection, let's say urinary tract infection or some other bacterial infection, you get an antibiotics for 10 days, 20 days, and that antibiotics get rid of the bacteria. So now I can call that you're cured from urinary tract infection or some other infection, because unless until another event happens where the bacteria comes and infect you again, that you can't get that disease again. That's what we call cure. That means there's no chance for the disease to come back. Okay. In cancer world, they also start to use cure now if the disease has been gone for 5 to 10 years, depend on different cancer. The reason is that if the cancer was going to come back, it would have come back in 3 years, 4 years, 5 years, or 10 years. The concept of remission is also from cancer world. That basically once you have taken care of the cancer cells, that means you have eliminated the cancer as best as you could, and you cannot detect any disease in somebody's body, then you say, okay, I think you're in remission. The reason you can't use the cure at that point, because still there's a chance for the disease to come back, because there could be some cancer cell hiding in some corner of your body for it to come back. Now I'm going to go to the immune diseases to explain the same thing. Immune diseases or all autoimmune diseases are our own immune cells attacking our own body or different parts of our body. So what happens is, and not the whole immune system is attacking our body, there's some bad players, and generally that bad player, it could be in 0.00001% of the whole immune system attacking the body. So when you calm down the immune system by immune suppressive drug or different drugs, what you do is you over the time cut down those bad players, or you make those bad players dormant. Then we say, okay, disease is improved because patients doesn't have any more symptoms, so let's call it remission. The reason again we cannot use cure is because that 0.00001% bad cells can become active again or can come back. They haven't gone away. We haven't killed the immune system and given you a new immune system, because it's still your own immune system, still your own genetic makeup, that cells can come back and cause the disease again. That's why the flares happen in autoimmune disease. You're very well aware of. Somebody could be completely doing well one day, one day can get a flare, or somebody is good for 2 years, can get a flare after 2 years. That's why we use the word remission again. Basically that means at this time point, I don't see any clinical disease activity in you, so you must be in remission, and that we can then cut down the treatment and get off. Maybe in 10 years, I have patients who have been disease free for 10 years. I say maybe I should use the word cure now, because if something would have happened, then but what is that time frame? We don't know. 2 years, 3 years, definitely not 2 years, because I've definitely seen many flare 2-3 years later. I used to think 10 years is my time frame, but I was proven wrong when a student came to me from CMU with dermatomyositis. I say, okay, I'm trying to explain him he didn't give me the history before, but I'm trying to explain him what dermatomyositis is and what happens and this. He said, I had it 12 years ago. Like, what are you talking about, 12 years ago? So I used to have cut off in my mind like 5 to 10 years, the disease can't come back and you cured. 12 years later, the patient had the disease come back. So that's rare. That's just one example to give you. Generally, 5 years is a good time frame. But then you can call it, at certain time point, you can start calling it cure. What's the time point is nobody knows. But our goal is generally remission. Thank you.